Let's now talk about the therapy for cancer. Drug therapy for cancer falls closely into four categories: chemotherapies, hormonal therapies, targeted therapies, and immunotherapies. So, chemotherapy is aimed at blocking DNA synthesis and cell division. Hormonal therapies interfere with the tumor cell growth. Targeted therapies such as small molecule inhibitors of cancer, and immunotherapy which induce or enhance the anti-tumor immune response. So this is our main concern, immunotherapies which will induce or enhance the anti-tumor immune response in our body. So what is cancer immunotherapy? Treatment for cancer by immunological approach is known as cancer immunotherapy. Reason for interest in immunologic approach is that most current therapies for cancer rely on drug that kills dividing cells or blocking cells, and this treatment have harmful effect on normal proliferating cells as well. Immune response to tumor should be specific for the tumor antigens and should not harm or injure normal cells. Therefore, immunotherapy has the potential of being the most tumor-specific treatment that can be devised. Cancer immunotherapy aims to augment the weak host immune response to tumors. That means active immunity, or to administer tumor-specific antibodies or T cells in the form of passive immunity. Immunotherapy stimulates patient's own immune system to harder or smarter to attack the cancer cells, so it has the minimal side effects on the normal proliferating cells. Types of immunotherapy. Immunotherapy can be two types: passive immunotherapy and active immunotherapy. So it is passive immunotherapy, administration of immunologically active materials or monoclonal antibodies which target either tumor-specific or overexpressed antigens. And active immunotherapy induced directly to tumor-bearing patients by cancer vaccines, cytokines, cell-based therapies, tumor-specific CTLs, tumor-derived APC, and dendritic cell priming. So what are the monoclonal antibodies used in cancer immunotherapy? Monoclonal antibody basically mark the antigen and attract other cells of the immune system to destroy the cancer cells. Monoclonal antibodies binds only on the cancer-specific antigens and induce an immunological response against the target cancer cells. Monoclonal antibodies can work alone, which is termed as naked monoclonal antibody. Then it works with conjugation of chemotherapeutic drug. It can be radio-labeled, chemo-labeled, or it can also act as immunotoxins. So what is naked monoclonal antibody? Antibodies that work by themselves. These antibodies bind to a specific antigen on the cancer cell and mark them. A person's immune system then work against marked cancer cells to destroy them. One example is trastuzumab, which target HER2, used to treat breast cancer and stomach cancer. On the other hand, conjugated monoclonal antibodies are those joined to a chemotherapy drug, radiolabeled particle, or toxins. Monoclonal antibody then delivers the conjugated substance to the target cell for proper action. Some example of radiolabeled monoclonal antibodies are ibrutinib toxetan and tocilizumab CD20AZ. Some example of chemo-labeled monoclonal antibody are brentuximab vedotin, which is a CD30 antigen, then immunotoxins like denileukin diphthitox. So how does monoclonal antibody works against the cancer? In one early success of the monoclonal antibody treatment, Art Levy and his colleagues successfully treated a 64-year-old man with terminal B-cell lymphoma that had metastasized to the liver, spleen, bone marrow, and peripheral blood. Because this was a B-cell cancer, the membrane-bound antibody on all cells had the same idiotype. That means antigenic specificity. So what is idiotype? The variable part of an antibody, including the unique antigen binding site, is known as idiotype. And what is isotype? An isotype usually refers to any related protein or genes from a particular gene family. Isotypes are distinct forms of the light or heavy chains which are present in all membranes of the species encoded at distinct genetic loci. Isotypes control are primary antibodies that lack specificity to target, and idiotypes are very specific to the target antigens. So these two terms is very important to understand the mechanism of action of monoclonal antibody on cancer cells. By this procedure, these researchers produced mouse monoclonal antibody specific for the B-cell lymphoma idiotype. So what is this procedure? First, B lymphoma cells isolated from the patients, which is then fused with human myeloma cells. After fusion, the hybridoma secreting B lymphocytes antibody are selected. Then these B lymphoma monoclonal antibodies injected from hybridoma into a mouse. Then the spleen cells from this mouse is isolated and fused with myeloma cells from another mouse. After fusions, this fused cell will produce anti-isotype hybridoma or antibodies specific for isotypic antibody determinants or anti-idiotype hybridomers. Then this anti-idiotype hybridomers, that is antibodies specific for the idiotypic determinants on antibody 1, are selected and transferred to, that means injected into patients, and this patient will then show increased immune response to tumor antigens. When this mouse monoclonal antibodies, that means the idiotype we have generated here in this experiment, was injected into the patient, it bound specifically to only the B lymphoma cells that expressed by that particular idiotype immunoglobulin. Since B lymphoma cells are susceptible to complement-mediated lysis, and the monoclonal antibody activated the complement system and lysed the lymphoma cells without harming others in these patients. After 4 injections with this anti-idiotype monoclonal antibody, the tumors began to shrink and the patients entered an unusually long period of remission. This is the development of monoclonal antibody specific for idiotypic determinants on B lymphoma cells. So let's summarize. Monoclonal antibodies generated using these procedures is specific for B lymphoma idiotypes in patients with terminal B-cell lymphoma. When this mouse monoclonal antibodies generated from B lymphomas from hybridoma is injected in these patients, it bound specifically to only the B lymphoma cells that expressed that particular idiotype immunoglobulin. Since B lymphoma cells are susceptible to complement-mediated lysis, the monoclonal antibody activated the complement systems and lysed the lymphoma cells without harming any other cells. So this mouse monoclonal antibody will be very specific to the target tumor antigen. After 4 injections with anti-idiotype monoclonal antibody, the tumors began to shrink and the patient entered an unusually long period of remission. Let's now talk about cancer vaccines. Majority used for treatment, not prevention, may offer methods that can enhance the immune response against cancer. This cancer vaccine can be several types. For example, tumor vaccines made from actual cancer cells that has been removed during surgery from a patient. Then antigen vaccines. These vaccines boost the immune response by using one antigen rather than the whole tumor cells. Then dendritic cell vaccines, special immunocells in the body that help immune system recognize the cancer cells. Finally, DNA vaccines. Vectors can be given bits of DNA that encodes protein antigens. Vectors are then injected into the body. This DNA might be taken up by the cells and can instruct them to make specific antigens. Finally, we'll talk about cytokine therapy. Cytokines are secreted and membrane-bound proteins. They have immunomodulating properties and therefore can be used as a therapy to active anti-tumor immunity. Cytokines can be interleukin or interferons, for example, interleukin 2 or interferon alpha. We will now discuss about this mechanism of action of interferon alpha and interleukin 2, and they used to treat advanced melanoma and renal cancer cells. How does interferon alpha work? Interferon alpha is secreted by nearly every cells in the body and involved in cellular immunoresponse against the viral infection. Type 1 interferons induce expression of MHC class 1 molecules on the tumor cells and mediate the maturation of the subset of dendritic cells. They can also activate CTLs, natural killer cells, and macrophages. Interferon alpha is the only currently approved adjuvant therapy for patients with high risk of stage 2 or stage 3 melanoma. What is adjuvant? Adjuvant is a substance which enhances the body immune response against particular antigen. Interferon alpha also approved for the treatment of some hematologic malignancies, AIDS-related Kaposi sarcoma, and as a component in anti-angiogenic combination regimen with bevacizumab for advanced renal cancer. Interferon alpha has dose-related toxicity and can lead to depression, confusion, mania, and other neuropsychiatric diseases. High doses may cause permanent alteration of immune system, causing vitiligo and hypothyroidism. So vitiligo is a condition in which the skin loses its pigment cells. So basically, interferon alpha induces the expression of class 1 MHC molecules on the tumor cells and mediate the maturation of a subset of dendritic cells, which can activate CTLs, natural killer cells, and macrophages and induce enhanced immunological response to tumor antigens. How does interleukin 2 works? Interleukin 2 plays a pivotal role in the treatment of patients with metastatic melanoma and renal cell carcinoma. National Cancer Institute found that adaptively transferred interleukin 2 activated peripheral blood mononuclear cells with administration of interleukin 2 in high doses. resulted in significant tumor regression in patients. It promotes both effector T cells and T regulatory cells, but its exact mechanism in the treatment of cancer is still unknown. The side effect for interleukin-2 may be capillary leak syndrome, which is characterized by hypotension, tachycardia, and peripheral edema secondary to third-space fluid accumulation. So what is tachycardia? It is a condition when the heart rate exceeds the normal rate in the resting phase. Side effects also include fever, chill, fatigue, gastrointestinal side effects such as nausea, vomiting, anorexia, cholestasis, and diarrhea. Interleukin-2 reported a 2% mortality rate. So how does interleukin-2 work? By promoting both effector T cells and T regulatory cells, and its exact mechanism is yet to fully uncover. That's all for cancer and tumor immunology. Thanks for watching.